Did the neuronal and glio populations differentiate the same as in the wild type mice?
Yeah, so what we think based on our results is that they differentiate differently in the mutant. You are talking every day to many different people, they have their different projects, and you feel that you are doing something for them. Why being a princess if you can be a scientist?
Welcome to the 2021 edition of Women in Histology, brought to you by the National Society for Histotechnology. This year's series features incredible scientists changing lives in the lab and making a difference to advance STEM within the community. In this episode, Andrea Transu sat down with Dr. Elisa Balducci to discuss her research journey with DYRK1A and to talk about what she's doing now that she no longer is working in the research lab. Also about her work with science outreach programs.
Hi, everyone. My name is Andrea Transu, and I'm the manager of the Histology Core for the Hermelin Brain Tumor Center at Henry Ford Hospital in Detroit, Michigan. So we work for principal investigators as well as the NIH and the Department of Defense. And we focus primarily on gliomas and specialize in using patient-derived xenografts for our studies. Today, I have the distinct pleasure of interviewing Dr. Elisa Balducci, who has extensive experience in biomedical research and biotech. She comes to the table with a wealth of project management skills, a business-oriented mindset, and a robust scientific background. On top of all of this, she is fluent in 3 languages and has a genuinely kind and gentle spirit. So Dr. Balducci, tell us a little bit about yourself before we get into your work with DYRK1A.
Okay, thank you very much, Andrea, and thanks everybody for listening. Yeah, my name is Elisa Balducci, and my background is a bit a mix of industry and science. It's also international. I am Italian originally, and I studied my university degree in biotechnology in Italy. Then I moved to Spain for the PhD in neuroscience, and then I had, let's say, a couple of years in the UK, and finally I came here in the US. Now I am based in Miami, Florida, and I work for Agilent Technology as a marketing program manager in companion diagnostics.
Nice, that's so incredible. So tell us about DYRK1A. Why this project?
Okay. So this started a long time ago, and it's actually very good to talk about that again, because it was an important part of my life. I started to work on DYRK1A when I started my PhD. I had just finished my university degree, and I was looking for a new challenge in research. So I had the opportunity to look at this laboratory. It was in Barcelona, and the PI was Marion Arbones, and she had a project with this mouse model that it's a model with only one functional copy of DYRK1A. And the reason why she was working on this model is because DYRK1A is in chromosome 21. So the triplication of this gene that codes for a protein kinase was associated with some developmental defect of Down syndrome. So she created this mouse, and it was very interesting for me, first of all, to see that the description of this mouse had a phenotype that was very strong. The complete knockout of DYRK1A was lethal, so it indicates that this gene was essential to life. And then the mouse with one copy of the gene had a very strong developmental delay and some autism phenotypes. For example, these mice, they didn't care about other mice, but they would spend a lot of time playing with the little things in the cage. So what was really interesting for me is, first of all, I love neuroscience, and I wanted to keep working on neuroscience. I did a project at the end of my university on Alzheimer's disease, so I wanted to keep working on that. And then what Mariona told me, the PI, is that she wanted to start looking at the differential gene expression of the brain of these mutant mice, because she wanted to understand really what are the drivers of this developmental delay. And my background was very molecular, so I was super excited to take this challenge. So we got started, and I started in our lab and continued all the project on DYRK1A.
Beautiful, beautiful. So how did the project start?
Okay, so we wanted to look at this gene expression in the cerebral cortex during development. So we decided to choose 2 developmental stages that are important in the development of the brain in mice, and they are P0 and P7. That is the day of birth and 1 week after birth. And we analyzed what the differences in gene expression between the wild type and the mutant mice. And in these stages, we know that the cerebral cortex, that is very important for the cognitive functions, it's actively differentiating. And it was interesting to see that at both stages, at P0 and P7, there were many genes that were differentially expressed in the mutant compared to the wild type. And there were genes of all types, of course. When you do these wide genome screenings, it's very difficult then to focus on something that makes sense, because everything looks interesting and you tend to start looking to something that you already know. So I found a group of genes that was interesting to me, and there were genes that are involved either in the glial differentiation or in the glial function. And when I say glial, I mean astrocytes and oligodendrocytes that are one of the 3 cell populations that, together with the neurons, that are generated by the neural progenitors. So the reason why I thought it was interesting is because usually when you see a developmental delay, it's easy to go for a group of genes that are involved in synaptic function or axiogenesis. But the glia usually is considered like a support to the neuron. So the astrocytes, they have like a repair function, and the oligodendrocytes, they create the myelin around the axon, so they allow the fast transmission of the messages between neurons. But usually they are not the main focus of the of study in neurodevelopmental defects. Even if there are like more and more indications that the glia has a role also in the brain function. And I think that Rett syndrome is one of the clear examples of this, where the glia has been associated with the brain function. So then is where I decided to follow that path and say, okay, now I want to see if in these mice there is a problem in the development of the glial cells, or the glial cells and the glial function.
So, okay, so you know that we have the differences in glio-related genes, but why is that?
Yeah, that was actually a good question. The first question, okay, the genes are altered. Now, what does it mean? It means that there is a change in the fate of the progenitors, so they differentiate differently in different proportions between neurons, astrocytes, and oligodendrocytes, or it means that the glial cells are dying more or less than the neurons, or maybe their function is impaired. So maybe the structural proteins like MBP in the case of oligodendrocytes or GFAP are expressed differently because it's the function itself that is impaired. So we had all these questions, and we started looking at the cell death first of all. So we discarded different situations because we did a preliminary study, and we decided to keep working on the question, are the progenitors differentiating differently in the mutant compared to the wild type? So we wanted to see if the cellular fate was affected, and then it could indicate that DYRK1A is in a way involved in deciding the fate of the neural progenitors. Do you have any question?
Go ahead. I'm actually excited. I'm just listening and excited to hear. Keep going.
it's also exciting to go back and tell the story again, because during when you do research, sometimes it's painful, but when you tell the story at the end, it's much nicer. So we wanted to look at the fate of the progenitors, and we decided to use an approach that was in vitro first, and then we wanted to validate the results in vivo. So for the in vitro, we had a system that was very nice. It's called neurosphere cultures. So we went to analyze the cerebral cortex of the mice. I think the embryonic stage was E14. At this stage, we consider the cerebral cortex made of progenitors, basically almost only progenitors. When we take those progenitors, you can put them in a medium that allows the proliferation of the progenitors. So these progenitors, they start proliferating and they create like a sphere that is it's a clone of the same progenitor. You can maintain these neurospheres for many passages. So in this way, you grow and you increase the population of the progenitors. Then at some point, when you have enough progenitors, you can decide to start differentiating. Instead of keeping them in a medium that allows the proliferation, you put them in a plate and you let them differentiate. And in this way, I remember we were leaving the progenitors differentiate for 1 week or 10 days. And after that, we started to use the markers for the neurons, for the oligodendrocytes, or the astrocytes. And then we started to count and say, okay, we had a population of progenitors in wild type and mutants. Now, are the differentiated population different in the two progenitors? And the answer was yes. So actually, we were, let's say, in the right track. We found out that the alteration of the genes that we had observed was associated to a different differentiation of the progenitors. And specifically, we were seeing more astrocytes and less oligodendrocytes. And this was cool. So then next step, we have to see if we reproduce the same result in vivo. And to do that, then basically we had to take the embryos of the mutant and the wild type during the embryonic stage. So we fixed the cerebral cortex and we did immunohistochemistry on the tissue. So we could see in vivo if the brain at this developmental stage had a different ratio of cell population. And again, we found the same results. So it was, everything was coming together. And I think this was an exciting moment of the work, because I was coming from a molecular background. So I was more about genetics. I didn't see anything. I didn't need to see any result with my eyes. I could trust the genetic results. But then when you are in your own project and it's years and years long, it's very nice when you see that the molecular part is confirmed in vitro, is confirmed in vivo. So everything makes sense together. And all these techniques are useful and they give you a piece of information. So that was very rewarding at that point. Yeah.
It is rewarding. So let me ask you this. Did the neuronal and glio populations differentiate the same as in the wild type mice?
Yeah. So what we think based on our results is that they differentiate differently in the mutant. So now we have a mouse with an altered population of neuronal and glial cells. And then the next step is what it's still ongoing, and somebody in the lab is still working on the continuation of this project. And how this affects the function of this population. And even if we want to look beyond, is how is this connected with the impairment of the cognitive functions of these mice. So that's why then we started some functional studies. And for example, when you look at the oligodendrocytes and you want to do functional studies, then what we did, was to look if the myelination was correct in the mutant as in the wild types. And this is one of the studies that are still ongoing. And so we were a good team. So my colleagues are still working in part of this project.
Nice. So as you were studying this, were there other things coming into the pipeline that gave you more or less information or drove the direction that you went into?
Yes. This was interesting. There were a few things. Well, there are many things coming up all the time while you do research. But I think the most important for us, for the whole lab, not only for me, was during this research project, it was described for the first time that there are humans that have a loss of function of DYRK1A. So it means that we were studying a mouse model for a human condition that hadn't been described yet. The initial idea was to study this model to understand better Down syndrome, but then we realized that there was a syndrome that is DYRK1A haploinsufficiency in human that has a very strong phenotype. It's autism spectrum disorder, and there is developmental delay, autism, everything that we were observing in the mouse model. This was super important for us because it kind of gave sense to what we were doing from a human and clinical perspective as well. And one of the things that have been described later on is the fact that the brain of these humans, they also had an increased astrocyte population. So there was an alteration in the glial population in the human DYRK1A heterozygous as well. So that was very important.
Okay, so this work that you're talking about has already been published, and I know that you are no longer with this lab, but are you still following, or do you still have your hand in anything going on right now?
Yeah, so a part of the study has been published, and there is another part that is more functional, that it's in process of publication. I am connected with the lab and with the project, and they are doing a great work to continue that. I'm very proud of the team.
That's still good that you have that ability, though, and it's good that you are following. I'm sure that your expertise is just immeasurable. So now that you're not there and DYRK1A is still your baby, but kind of in the teenage, going away to college stage of its life, what's next for you?
After like 5-6 years, I left the lab because my PhD came to an end, and I was looking for a new challenge, and I moved to the industry. Mm-hmm. And the reason why I moved to the industry, because I really enjoy the fact that when you work in the industry, you can help many scientists instead of just being concentrated in one project, one gene, one protein. You are talking every day to many different people. They have their different projects, and you feel that you are doing something for them to help achieve their objectives, their scientific objectives, and you provide the technologies. So that was the main motivation to move to the industry, and I'm still in the industry, so I like it, apparently. And so first I worked more in genomics, and now currently with Agilent, I'm working in companion diagnostics, which is it's closer to the clinical outcome of research, because you see directly how a biomarker can affect the treatment of patients. It's exciting to see how people work so hard, really giving a better life to patients of all type. I worked in neuroscience, now I work more in cancer, but the bottom line is that the scientific community is great, is amazing, and everybody is doing something that maybe it looks to drop in the ocean when you work on it, and sometimes it's very frustrating because you don't see the result that you are expecting, or you don't see why certain things certain results are as they are. But then when you see the whole picture, it's incredible what the community can achieve.
I can imagine that both of these were very rewarding. And I can see that your expertise is well used in both aspects of the field. But I really want to know which one you like the most and why is that?
This is an interesting question. I think I like being in the industry because it reflects probably my personality, my mindset. It gives me the opportunity to interact with more people and to have results quicker than in science. This is, but I have to say that the people that I've met during my scientific career when I was in the lab, the people are amazing. It's just, I'm so proud that all my friends from the university, from the PhD, they are still working so hard to give answers to problems that we face, that patients face. I enjoyed being in research very much.
I can understand, though, the difference because research is definitely the long game. Some investigators work on a project for the span of their careers. I know one researcher who's been working on this same gene for 20 years now. And so I can understand how that is rewarding eventually, but I understand how that reward is really far off sometimes. So in all of your studies and in all of your travels and every place that you've worked in and everything that you've done, what have you learned?
Yeah, I think I've learned that for me, that science is my passion. And I understood that I can work in different aspects of science, being in research, being in industry, but it really motivates me. And I wake up happy every morning when I know that I'm working with scientists, that I have a purpose, that it makes me very happy. So I realized clearly that was my path. And then I've also learned that sometimes changes are very difficult. I moved from Italy to Spain, and then from Spain to the UK, and then the US. And every time you change, it's hard. It's hard to change the topic of your job, but also to change country, and you have to start everything from the beginning. But at the end, every time you learn something new, and suddenly you realize that you have a baggage with you. And it is thanks to all these different experiences. And if you give the best that you can and you work with passion, you want to learn and you want to change the world, because this is something that I also like. I want to think that I can change the world with the things that I do. And then at some point, there is some reward for you emotionally and intellectually.
Absolutely. Finding a purpose, finding a gift that you can give to the world that's unique to you and what you have to offer, and that passion, it really is a beautiful thing. And we need that, especially in science. You really need passion, because if not, then it's really pointless. It doesn't do anything. All the facts, all the figures, all the science that we know and understand, it really doesn't mean if you don't have a heart for it. And you've really got to have a heart for the lab.
One of the reasons why I moved from the academia to the industry. When I was in research, I started a non-profit organization with other researchers, and it was for science communication. And I suddenly found this passion for letting everybody in the society know what science is about. Because especially in Spain, where I was working at this time, the citizens, they pay research with their taxes, and they deserve to know how scientists spend this money and why it's important and what we do, because it's something that sometimes is very far away from your understanding. But it's not, because your health, everything, you need to know something about what's going on in your body, what is a disease, what are certain mechanisms that are behind this. So we started doing these kind of things like science cafes in different cafeterias in Barcelona. And it was very nice because maybe we have like 50, 100 people, all ages, and they were just interested to understand what is personalized medicine, why vaccines are important, all type of different topics, and asking questions. And I I felt at that point so happy when I could explain science to other people. So we started also to go to schools and high schools, and we were doing like workshops. But it was nice to see how young people were excited when they were owning their project, and they say, okay, I have this question. How can I answer this question? How can I apply the scientific thinking? Because this scientific thinking is something important in life in general, to think properly what are the causes of something and what are my controls when I think about something. It was good to see how young people could appreciate all this process in a small activity. And I think all this contributed also to my choice to say, I want to do something that is a bit more social. I want to talk to other people about science. And that's why I ended up in the industry as well.
That's awesome. So let's talk about, because this is a women in histology show, so we can talk about this. Let's talk about how your classes really affected the little girls. How did you, was this a whole new world for them? Did you see some girls that came up to you and said, hey, like they could be the next scientists, they could be the next anything? Did it open up their eyes to the new possibility that they, as young women, could be scientists and make really impactful differences in the world?
Yeah, this is good. Yeah, there were little girls coming and asking questions, and I was so proud of them. Like, please keep doing this. And recently, I read this sentence. It's not mine, but now it's mine too. And it was saying, "Why being a princess if you can be a scientist?" And it's true. There are things that are so exciting in science, and I still think that a lot to do for women in science. So I really encourage all curious little girls to go into this path. When I was young, my female, like the woman that I wanted to be, was Rita Levi-Montalcini. She discovered the neurotrophic factor, and she was an Italian Jewish during the war, and she did her research like hidden, and she found the neurotrophic factor like something huge. And I've always thought, I want to be a woman like that. I don't want to be a model or anything else. I want to be a woman like that one. So I hope that there are a lot of girls out there who want to pursue this career.
That's awesome. And so by that, you learned that representation matters, right? Because you knew you could do this because you saw her doing it. And so now you're doing it, and now other little girls can see you doing it, and they know that they can do it. And so number one, what is the name of your nonprofit? And number two, is it local?
So the name of that nonprofit association is Biocomunicat. It's like a Catalan name that is the language that is spoken in Barcelona. It means communicate biology. Then we have a webpage, and of course, I'm not active in this group right now because I am in the US. Even if I would like maybe to do something similar here, it would be amazing. But they are still working actively in the Barcelona area.
Wonderful. So, here in the US, we have this big STEM initiative going that puts science at the forefront of knowledge and, science and math and technology. So have you considered beginning a program or expanding your program here? And you could work with the local school systems, you could work with the NSH, you could work with really any agency, and you kind of have that platform. Would that be a passion for you? Would that be something you'd want to explore?
It would be a passion. Thank you for bringing this up, because it's so easy to keep doing what you are doing every day. And sometimes you have to think again in your passion, when you have a family and kids and things like that. But this is something that really motivates me. So I have to look into that again here in the US.
I'm really looking forward to seeing that. I'm exploring, my boss and I actually have the same passion about introducing STEM to girls all over the world. And so she recently traveled to Liberia to do the exact same thing. And so you have these areas where women and girls are not taught the same way, and they don't have that same access to education. And so women like you serve to show girls around the world things that are possible. And so it's just so important to get that out there. So I'm really encouraged by your passion. So is there anything else that you want to tell us?
No, I just can say thank you, because it was very good to talk about my project again, and also talk about my passion once more. And it's important. Thank you for giving me this opportunity.
Thanks for listening to this episode, and don't forget to check us out on your favorite podcasting site. Use the word HistoTalks. Awesome podcast!